So welcome to the second day of the RNA Cozy. We'll start now so that we stay on time for the day. So this morning our first speaker is Chen Yang from the BC Cancer Genome Sciences Centre in Canada, and her title is her talk is titled "Is This the End? Terminator Transcriptome Annotation with Deep Learning." Where I'm supposed to go? To view. Full screen. Oh, slideshow, yeah. Thank you.
So, hi everyone, my name is Chen. I'm a grad student from the University of British Columbia and also and also BC Cancer Agency Genome Sciences Center. First of all, I would like to thank ISMB, ECCB, and also RNA Cozy for having me here to talk about my work today about transcriptome annotation with deep learning. So why is it important? First, let's look at a schematic diagram of a mature mRNA. So a mature mRNA is starts from the 5' cap, and then the transcription start site, and then the 5' UTR CDS, and then 3' UTR, and followed by the polyadenylation site with a poly tail. Although the 5' UTR and 3' UTR, they are untranslated regions, they still harbor extensive regulatory sequences such as microRNA binding sites, RNA binding protein motifs, and they have very important roles in transcription regulation and translation regulation. So if we can precisely locate the 3' end and 5' end, we'll be able to identify novel isoforms, and we can also perform functional analysis on those elements. We may even be able to find new elements. And with these two combined, we can perform more regulatory analysis and differential expression analysis to uncover the myth of gene regulation in various biological processes, such as in cancer. So generally, there are three steps involved in the termin annotation. First, the sequencing, and then depending on the sequencing technology we're using, assembly may or may not be needed, and then the annotation. So generally, there are two types of sequencing technologies. One is 5' and 3' and targeted sequencing. With this type of sequencing, we can precisely locate these sites with very high sensitivity. However, these technologies come with they are more costly and they are more technically demanding. And they have a relatively less broad applications other than detecting the sites. And for the poly side, these technologies also suffer from internal priming. On the other hand, RNA-seq, regardless if it's short read sequencing like Illumina or if it's long read sequencing like PacBio and Oxford Nanopore, they have very wide, broad applications other than simply termin annotation. They can also be applied on for transcriptome, for differential expression analysis. That's why they have become the standard protocol in transcriptome analysis. However, when it comes to the termin annotation, they will have less sensitivity because of the uneven coverage in UTR regions. So further analysis will be required, and specialized bioinformatics tools are needed. In fact, there are a lot of bioinformatics tools have been developed already. For example, for the TSS detection, tools generally fall into two categories. One is deep learning based methods. However, these methods, they generally focus on predicting the promoter regions instead of the TSS, the precise site. And homology-based search methods like maker and breaker, they have relatively long runtime, and they are based on the conservation of between species, but the conservation levels may fluctuate across the genome. For the poly-A CS detection, on the other hand, there are 3 mainstream approaches. Like the deep learning ones, they generally focus on the poly-A sites associated with poly-A signal, which is the hexamer. However, not every poly-A CS are associated with one. So ignoring those sequences may affect the sensitivity as well. And then there are a read evidence-based approach represented by CLEAT, and also expression level-based approaches represented by the parse. These approaches, they are heavily influenced by the library preparation and read coverage. So here in this work, we present Terminator. It's a completeness assessment pipeline for transcriptome annotations. So in this work, we simply ask one question: given a transcript context, are their ends the real ends of a functional transcript? So we start from raw RNA-seq reads. If you have Illumina reads, you probably need to do de novo assembly. If you have PacBio reads, that step can be eliminated. And then starting from the transcript context, if you have reference genome, you can align it back to the reference genome, so you'll have extra information. If you don't, we'll simply take all the 5' ends and 3' ends of the transcript context. Those sequences will be our candidate sequences containing potential TSS and potential poly-A sites. Then they will be fed into our deep learning model to determine if those sequences actually contain a TSS or not, or poly-A site. If both of them are yes, then yes, this transcript is complete. So here's the core of our deep learning model. In the training stage, we start from the reference transcript. We align it back to the reference genome to extract the flanking sequences around the target sites. And then the raw sequence are represented by a sequence of k-mers. And we are using 4 different Ks here. All the k-mer representation of the raw reads are concatenated together, and they are one-hot encoded into, and they will be the first layer of our model. And then going through the embedding layer, each k-mer, regardless of its length, they will be represented by a vector of length 128. And then followed by 3 fully connected layers. All the weights are collapsed into one single probability, telling you whether the sequence has a TSS or not, or has poly-A CS or non-poly-A CS, or simply non-CS. I'll explain about these 3 levels later. So for the Fiveframe model, we generated our data sets. We generated two data sets in total. One is human and one is mouse. So we are using specialized databases like DBTSS and EPD-New, because these databases catalogs all the TSS with experimental evidences. And that's how we identify the true TSS. And for the negative ones, we simply extracted random sequences from the genome as long as they are 100 base pair away from the true ones. And we are extracting them. They are equally selected from 5 different types of regions across the genome, like the 5' UTR, exon, intron, 3' UTR, and also intergenic regions. So similarly, for the 3' model, we are doing similar things here, but I want to briefly mention why we are having this non-poly CS. That's because when we were trying to map all the poly sites in these databases back to ensemble annotation, we found that some transcripts has no poly sites identified on them anyway. So we are just simply taking their ends and label them as non-poly CS because they have no evidence. And later I'll show you how adding this label is helping us to improve the accuracy. So then we performed an extensive hyperparameter combinations search to optimize our our model through five-fold cross-validation. And here we can see, so here it's simply showing how our model is able to group different group sequences with different labels separately. So here we're taking the last hidden layer of our model, all the weights of all the sequences, and performed sequence clustering. And then we superimposed the real sequence labels onto this clustering to visualize. So here we can see the different colors have quite good separations, especially for the Threeframe model. You can see there's the little yellow cluster, which is representing the non-poly A CS. And they are actually separate from the other two classes. So from now, I'll focus on the Threeframe model because of time limit, but we did everything the same on the Fiveframe model as well. So there are a lot of regulatory motifs and sequences around the poly A sites, but we don't know how long the sequences are actually needed to be to have an accurate prediction. That's why we constructed 40 models with different upstream and downstream length combinations. So here on the upper panel, you can see each facet represents one downstream sequence length. And the x-axis in each facet represents the upstream sequence. So the very first data point here represents the model built with only 20 nucleotide upstream sequence and no downstream sequence. It's still able to give a pretty good prediction with accuracy over 87%. And because if you look at the panel lower panel here, there's a very strong signal within 40 nucleotides upstream of the real poly A site, and consistently in our model, we found that as long as you have more than 40 nucleotide upstream sequence and more than 20 nucleotide downstream sequence, you'll be able to get an accuracy of over 94%. And actually, the sequences are not the longer the better. They start to saturate at that point. So next, for illustration purpose, we built our model with 100 nucleotide upstream and 100 nucleotide downstream. And we built this model with a human dataset and also mouse dataset. The reason we built mouse dataset is because we want to see how the information learned from one species is able to be transferred to the other species. So we applied our pipeline on the universal human reference RNA-seq data. It's a Illumina library, which is poly selected and stranded. So we compared our results with 2 other deep learning models using convolutional neural network, and we also compared it with CLID, the read evidence-based approach. We can see that the navy color curve, which is Terminator built on human, it's outperforming all the other competitors in terms of sensitivity and precision. By the way, the ground truth here is all ensemble annotated transcripts. But we all know that not all ensemble annotated transcripts are expressed at the same time in one library. So here we are taking all the expressed ones. They are quantified by Salmon, and we apply different TPM cutoffs to see how well our models is able to predict the expressed ones. And it turns out that in all 4 tiers, Terminator is able to outperform all the other tools. And especially with the TPM cutoff over 20, the Terminator model built on mouse is able to outperform the other 2 deep learning models as well. So the last question we want to ask is, since our model works so well on Illumina reads, how does it work on PacBio reads? So here we took the NA12A78 sample from Genome in a Bottle project because this dataset has this sample has matching PacBio and Illumina libraries. So we applied our model on these two libraries, and it turns out that our model works even better on PacBio reads. You can see the sensitivity is always twice or more than Illumina. And also, as we are getting more and more stringent on our probability cutoffs, the overlap between these two libraries are getting higher and higher. So here to summarize, here we present Terminator. It's a deep neural network-based pipeline. It is able to assess the completeness of transcriptome annotations. It can accurately predict the completeness of the 3' end and also 5' end. It can be applied on both short-read sequencing and long-read sequencing technologies. So it will help us to identify more novel isoforms and also facilitate functional analysis at isoform level. So last, I want to thank everyone, all my colleagues from the Bioinformatics Technology Group, led by Dr. Inaj Biro. And this work is funded by Genome BC and Genome Canada. And I also like to thank our bioinformatics program from UBC and also from the RNA COSI to help me support my trip here. So next, my colleague is going to present his work on de novo assembly using ONT RNA-seq data. And there are also a few posters hanging out there until 2 p.m. If you're interested, go and check. Thank you. I'm happy to take questions.
Hi, so nice talk. I have a question with regards to sensitivity towards measuring differences in APA. Could you tell us a little bit about it? I mean, what levels of alternative polydenylation do you need to see to sort of figure out differences, let's say, between two different conditions?
Well, here we're just recording the cleavage site we identified. It's not alternative polydenylation.
So your algorithm identifies a potential sequence, okay, without sort of giving any information about sort of use of it compared to another site?
No, it's simply based on the sequence information. Given a sequence, is there a site there?
Can you start to use it to sort of develop something that allows you to determine differences in the levels? Because that's really what the field needs.
Right, yes. So, well, because we are able to profile all the poly sites within a given sample. If you have two samples, we can quantify both of them, and we can identify if there's any novel sites found in one of them but not the other. And also if one site is found in both, we can also perform like differential expression analysis simply on the 3' UTR to determine the APA. Thank you.
Hello, here. So it's not exactly the same, but it's a related question. So you have been showing how this performs with PacBio, and I assume that you are using like already assembled transcript sequences of process transcript sequences, and PacBio really selects the reads to have a poly-A tail. So the polyadenylation side is kind of given already. So my question is, with your method, exactly how many of which percentage of the PacBio reads you are really discarding as not having this feature? Because I would say that the vast majority of them have true polyadenylation sites. So that's one question. And the second question, you have been showing the 3' end site, which is a kind of the easy one, right? So how do you perform predicting the 5' end?
Okay, thank you. So to answer your first question, our Illumina dataset is also poly-A selected. It's having a lower sensitivity because of several reasons. But for the PacBio, we do notice that there are higher percentage of sequences containing the poly-A tail comparing to Illumina reconstructed transcripts, but not all of them. Does that answer your question?
Yeah, but I mean, at the end of the day, which is the percentage that you discard as not having a true 3' end? So are you? I mean, if all of them, if 90% of them or 95% of them are okay, so
Yes, I understand that. I don't remember the exact number now, but we have already submitted our 3' and work to bioarchive, and in that manuscript, we have the exact number. And to answer your second question, the 5' end, we actually have a better sensitivity than the 3' end. The reason I'm not showing that here because we can't really find a competitor to compare with. Not like the 3', we have 3 different competitors, so it's easier to explain here.
Okay, thank you.
Thank you.
One last short question.
Hello, I found your talk very interesting, and I would like to ask how you deal with the problem of internal priming, because I heard that you are selecting random sites as the false sites, but if you're really randomly selecting, then you will probably only get 1 or 2 internal priming sites in a million. So, is your database really well trained to filter out internal priming sites?
Well, first of all, we are not trying to deal with the internal priming problem because in our training set, one sequence contains only the potential site in the middle and the upstream sequence, and the downstream sequence is actually genomic. It doesn't have any polytail in it, so we don't have to deal with the internal priming issue.
Okay.
Yeah, thank you.
Thank you.
Thank you.